Today, we're going deep into the world of microscopic warfare. We're going to explore one of the most incredible dramas in all of biology, how a tiny, tiny virus can completely take over a cell that's thousands of times its own size.
I mean, think about that for a second. How is that even possible? A virus is, well, it's basically just a little bit of genetic code wrapped in a protein shell. It doesn't have a brain, it doesn't have muscles. So how in the world does it pull off this amazing act of biological espionage?
Well, the answer is both elegant and, honestly, a little bit terrifying. The virus doesn't try to fight the cell head-on. No, it rewrites the cell from the inside out. It's a genetic coup d'etat. It seizes control of the cell's own internal machinery for one single purpose: to make more viruses.
Okay, so to really get how this works, we need to meet the key players that the virus is targeting, because this is all about reprogramming the cell's core operating system. First up, you've got this thing called RNA polymerase. The best way to think about it is as the cell's master factory machine. It just chugs along the DNA blueprint, reading the code and building everything the cell needs to live. It's incredibly powerful, but it doesn't really know where to go on its own.
And that's where the sigma factor comes in. This is the real key. If RNA polymerase is the machine, the sigma factor is the pilot. It's this little protein that latches onto the big machine and tells it exactly where to start reading a gene. So by changing the pilot, the cell can control which set of instructions get read. And that means if you can control the pilot, you control the entire factory.
And this, right here, is where viruses get so incredibly clever. Why build your own factory from scratch when you can just walk into someone else's and swap out the pilot for one of your own?
A perfect example of this is the battle between the SP01 virus, that's a type of virus called a phage, and the bacterium Bacillus subtilis. This phage pulls off a perfectly timed three-phase attack, and it does it by systematically changing that pilot, the sigma factor. Okay, get this, because the way this unfolds is just brilliant. It happens in these perfectly timed waves. First, the virus gets in and cleverly uses the bacterium's own pilot to read its early genes. But one of those early genes contains the instructions for a new viral pilot, a protein called GP28. This new pilot then kicks out the old bacterial one, hijacks the machine, and forces it to read the virus's middle genes. And what do you think those do? Right? They create a third set of pilots, which take over and direct the machine to the late genes. And those final genes, they're the instructions to build hundreds of new viruses and ultimately burst the cell open. It's a beautiful, cascading failure of the cell's defenses.
But what's so wild about this is that this strategy, switching out the pilots to run a new program, isn't just a viral trick. Bacteria like B. subtilis use the exact same mechanism to save themselves. When things get tough, like when they're starving, they swap out their own sigma factors to run a totally different genetic program, one that turns them into a tough, dormant spore that can survive for years. It's the same tool, just used for totally different reasons. One for a hostile takeover, the other for survival. But not all viruses are just these simple smash-and-grab artists. Some are way more sophisticated. Let's take a look at phage lambda. When this virus infects a cell, it doesn't just blindly attack. It actually makes a decision. It gets inside, and it has to choose between two very different paths. Path 1 is the lytic cycle. That's the all-out assault we've been talking about. Replicate fast, burst the cell, get out. But then there's path 2, the lysogenic cycle. This is the stealth approach. The virus can literally weave its own DNA into the host's chromosome, becoming a silent passenger called a prophage, and just wait. It gets copied for free every single time the cell divides, lying dormant until conditions are just right to strike.
So how does it decide? Attack now or hide and wait? Well, it all boils down to a molecular showdown, a race between two competing proteins that the virus itself makes. Whichever one wins determines the fate of the cell.
On one side, you have a protein called Syk. Let's think of Syk as the guardian. Its entire job is to shut down the viral attack and keep everything in that quiet, dormant, lysogenic state. And on the other side, you've got the CRO protein. We can call CRO the executioner. Its job is the exact opposite. It wants to push the virus into the lytic cycle, full-on replication and the death of the cell. So let's say the guardian wins. How does it pull it off? Well, the CI protein binds to a couple of key spots on the viral DNA.
And this does two brilliant things at once. First, it physically gets in the way, blocking the gene for the executioner, CRO, from even being read. And second, at the same time, it turns on its own gene, creating this positive feedback loop where it makes more and more of itself. With the executioner silenced, the virus goes dormant. Lysogeny is established.
But what if the executioner wins the race? CRO's strategy is all about speed and priority. It has a favorite binding spot on the DNA, one that it can grab onto first. And by binding there, it completely blocks the gene for the guardian from being made. It shuts Chi down before it can even get started. And with no guardian to hold it back, the viral assault machinery roars to life. The cell is toast.
Okay, but wait a minute. This whole thing begs a really big question. How can these two proteins, the guardian and the executioner, be so precise? How do they know exactly which spot on the DNA to grab onto? Well, the secret? It's all in their shape.
It turns out many of these DNA-binding proteins share a really simple but beautiful structure. It's called the helix-turn-helix motif. And it's pretty much what it sounds like. It's two little helical sections of the protein that are joined together by a sharp turn. Just a wonderfully efficient piece of molecular machinery.
And here's the magic. One of those two helices, they call it the recognition helix, is shaped to fit perfectly inside the main groove of the DNA double helix. The specific bumps and ridges on that protein helix, the amino acids, line up perfectly with the specific chemical signature of the DNA sequence. It is quite literally like a molecular key fitting into a genetic lock. And that's what gives it the incredible specificity it needs for this whole battle to even happen.
All right, let's zoom out for a second. We've seen how viruses can swap out a cell's pilot, and we've seen how they have these molecular duels to decide a cell's fate. What does this all really tell us about life itself? Because you have to realize what we've been looking at, these aren't just weird little tricks that viruses use. These are the fundamental rules for how life processes information. This is biological circuitry. This is how genes get controlled, how complex programs get executed at the molecular level in all of life. What we're really looking at here is genetic software. The DNA is the hard code, and proteins like sigma factors and CIN and CRO, they're the programs that run on it. Even these simple organisms are using this incredibly sophisticated software to make complex life-or-death decisions based on nothing more than the cold logic of molecular interactions. And that just leaves you with this one kind of mind-blowing thought. If this is the level of sophisticated programming that's running inside a single bacterium fighting a single virus, just take a moment and imagine the sheer complexity of the genetic software that is running right this second inside every single one of the trillions of cells that make up you,